So as I said earlier, but the good news is you do not have to spell these two method names, right? That's a matching question or multiple guess on the test, right? But you do need to be able to recognize them, right? So for acid-fast staining, right, in your book they have two different methods, right? But we're not using the Quignon method. We're using the Zeeland-Neeson method, where we're using our steaming pots. The endospore stain, we're using the Schaefer-Fulton method, right? It's important that you know that because there are other methods out there that will give you different results because they use different stains. And there's a distinct procedural difference for the Quignon method. The carbofusion that's used is different. It's more concentrated for the Quignon method, which is one of the reasons why we don't do that. It has to be shipped to us biohazard. And it has a higher content, I think, of the phenol. And so when you guys are doing your acid-fast stain today, you'll smell this smell. It's the phenol that's in this dye. And so you definitely don't want to sit over it and inhale the vapors, right, from it. It's not good for you. But that one would be really bad. You would have to do that in a fume hood. And as you can see, we don't have that one. We don't have a fume hood in this lab, right? So we don't, that's one of the main reasons why we don't do the Quignon method. And we use the Zeeland-Neeson method instead. Because we don't use that higher concentration of the carbofusion, you definitely have to use the heat. But I find that I've used both methods. I find the heat method is the best method. And when I worked at Hansen's Disease Center, they always used the heat method for making their slides. They came out best out of the two methods. Two most commonly used methods anyways. I'm sure there's probably more out there. So using this particular method, pretty much any of the methods, so you're still going to get the same color because it's still the same primary stain you're using, the carbofusion. It's just different formulas of the stain. So you'll see this characteristic clumping. The ones that do stain and stay stained in this procedure we refer to as acid-fast bacilli. They are rod-shaped for the most part when it comes to the bacteria. And they do tend to clump together, and this is mostly too because, remember, a lot of times we use water to create our motion on our slides. And these components of the cell wall for mycobacteria are very waxy. They don't like water. They're hydrophobic. And so that causes the cells to clump together. So that substance is called mycolic acid. And these guys have this in addition to the peptidoglycan layer. So notice they have the peptidoglycan, right? And then they have this additional sugar layer, and these mycolic acids are the very outermost covering for these guys. And lots of carbon associated with it. Where phospholipids typically have about 12 to 20 carbons, these mycolic acids can range from 20 to 26 and all the way up to 50 to 60 for the longer chains. So really big molecules. These organisms, if you were to gram stain them, they would come out gram positive. But a lot of times a relatively light purple because it's really hard to get the dye into these cells. And so one of the ways that we help dye these cells is that we heat them up, right? And what happens to wax when you heat it? It melts, right? And anything stuck in that wax, right? If you melt a candle, right, and you have liquid candle wax and you get something in it and then that hardens, forget about getting it back out, right? And so the similar thing is happening here is that we're heating them up, they're melting, the dye's getting stuck in that melted wax and it stays stuck there. So why would we want to know about these types of organisms? Well, a lot of them are pathogenic, cause disease, right? So it's an important way of identifying them. So as I said, I worked at Hansen's Disease and studied leprosy in the laboratory. And they do have, outside of Baton Rouge and St. Gabriel Parish, in an area called Carville, they still that's where the old leper colony was. There's still a couple patients that still live there and have, unfortunately, some of these deformities. It is a bacteria. It can be treated by special antibiotics now. But before the advent of that, it was highly stigmatized. People were sent away from their homes to live in these leper colonies. There's an individual there where he has deformities and losses of some of his fingers from the disease before the drug treatment came online. The other thing is that this particular bacteria really preferentially targets your peripheral nerves. It likes that cooler regions of the body. And so it would cause nerve damage, and we really can't fix that, right? We can kill the bacteria, but we can't fix the damage that it caused, right? And a lot of times, too, that with diseases, that's really what kills you, right, is the damage that it causes to the body. So really cool place to go check out if you're ever outside of Baton Rouge, heading to LSU, right? Something like that. It's treated on an outpatient basis nowadays. There's still about 100 new cases in the United States each year. It's not highly infectious, although armadillos do carry it, so leave them alone. Don't eat them, don't slaughter them, leave them alone. Don't call the Hansen Disease Center and say, come get my armadillos. They won't. And one armadillo will destroy your entire garden. Looking for insects. They're insectosaurs. And that's why they're also referred to as Texas road speed bumps. Because they go out on the road at night to feed on insects. They don't see real well. They are mammals like us, so they have hair, and the car's rushing by. It scares them. Well, their immediate reaction is to jump up and run straight in the direction in which they were facing, which is usually up into the car. And so, you can't even try and avoid them if you want to. So, interesting little buggers. So they study them, as well as they use mice, of course, for studying the disease. And a lot of the, because of the nerve involvement, a lot of the advances that they've made with leprosy research has actually helped out diabetics that deal with nerve injury due to the disease of diabetes. So, serious problem in India. Still a pretty serious problem in India. Another one that is of concern for us is Mycobacteria tuberculosis. This is, of course, much more widespread. And the reason for that is much easily spread. And the scary thing is that our immune system isn't able to get it under control. It can hide in our phagocytic cells and continue to grow in our lungs. As little as 10 bacteria can cause you to have this infection. We do not vaccinate against this in the United States. The main reason for that is the early detection system we use, which is the TB test, would no longer be effective. Once you've been vaccinated, you'll always be positive for the TB test because it's testing to see if your immune system recognizes the proteins associated with this bacteria. So some of you guys that are going into the allied health, nursing type professions, depending on where you work, you may run into a situation where they give you the TB test potentially every 6 months. Over time, if not done properly or just because you've had it done so much, you might end up with a positive TB test because the TB test itself may have vaccinated you. May have been enough of the introduction into your body of that protein that your immune system recognizes it. Once your immune system recognizes it, it's pretty much going to always recognize it. So once you have a positive TB test, you'll always have a positive TB test. So you'll have to have chest x-rays done to check, right? There's early detection. This disease is very slow progressing. In old times, it was referred to as consumption, right? Over time, the people just couldn't breathe anymore because the infection had taken over their lungs, and they would suffocate to death.
The TB test itself, when they inject you, and it makes the antibiotic, how does that work?
So what they're doing is they're just injecting some of those proteins into your skin, and so it's your immune system, if it recognizes, is going to respond, and so you'll get a bump if your immune system has responded. And it's a relatively slow response because it's a cellular response, so that's why they check it like 2 to 3 days. It's not like within minutes kind of thing. Unlike allergic type reactions where some people it's literally within seconds that you would get that response. So again, don't freak out, and then you know, there are lots of people that, you know, have been vaccinated that have come from other countries and stuff like that. So, you know, you get a special little card once you've got a positive TB, right? And there's some reasonality. But, you know, if you haven't worked with a patient, right, recently or something like that, that had it and you have a positive, no reason to freak out, right? Positive TB test just means your immune system is recognizing it, right? It doesn't necessarily mean you have the infection, but it could. And that's why it's helpful, because we could detect it much earlier than you would be feeling symptoms. Does that make sense? Okay. So, other ones, and this is just FYI, and these are mentioned in your book, right? Some other pathogens that can stain acid fast, right? But when it comes down to bacteria, we're mainly looking for the two pathogenic ones. We're looking for leprosy and tuberculosis. So I'm not going to go over these, right? These are there for you. This isn't actually even an acid-fast stain, but there's some eukaryotic parasites that they'll use the Endospore stain to help, acid-fast stain to help identify them. So as I said, right, in the Zealand-Neeson method, we're going to use our steaming apparatus. You can fit, as you can see, 4 slides on at a time, right? Since we have a pretty full lab today, we'll be packing these. Your filter paper is placed on top of your slide to keep it moist, right? You want to keep it moist through the whole procedure. Add dye as it starts to dry out. And definitely wear your goggles. That's going to take the longest time, right? We're going to leave them on there for about 5 minutes. So why do we say we use the heat? To melt the waxy substance, that mycolic acid in their cell wall, right? Helps get that dye to really stick in there. Are the acid-fast negative cells stained by the carbofusion? Yeah, just like with our gram stain, right? What are we going to do to remove the color? We're going to decolorize. There's a special decolorizer for this procedure. It's acid alcohol. And that's where this procedure gets its name, acid fast, right? So despite using acid alcohol, it's actually 3%, I think, hydrochloric acid. This is a very strong acid. This is a very strong decolorizer. So you're all like, oh boy, right? The good news with this, because of the heat and everything like that, I've never seen anybody over decolorize an acid fast stain, right? This one you can keep dripping until it's clear, and you're not going to decolorize your acid-fast organisms, right? So that's a very strong, as I said, decolorizer. So despite that, though, the acid-fast organisms stay stained, right? So that's what we mean. Acid, right? Fast, meaning that they stay colored despite the use of the acid. Now, at that point, our non-acid fast ones, would you be able to see them? No, they'd be colorless. So you've got to counterstain them some contrasting color. Our procedure calls for methylene blue, right? Why do you suppose the acid-fast stain is not as widely used as the gram stain? Yeah, very few organisms are actually going to come out positive. All the rest, as I demonstrated in our staining ID, what do we keep saying over and over again? Non-acid fast, non-acid fast, non-acid fast. Right, so when we give you guys your unknown organisms, right, if you happen to be the one person that may have gotten mycobacteria and do the acid fast stain, bam, you've got it, right? But if you have one of the other 16 other organisms we have in lab that we could potentially give you, it's not going to tell you much, right? It's going to be non-acid fast. Where if you do the gram stain to start, if it comes out gram positive, right, and you have that clumping and they're rod shaped, then you're going to be like, oh, I better do the acid fast stain, right? It's probably acid fast. Maybe I got the mycobacteria. Other than that, when you do the gram stain, it's going to pretty much divide your possibilities in half. About 60% of all the known bacteria out there are gram negative, about 40% are positive. So it'll help narrow down your field basically in half of what you're potentially dealing with. So the problem is that, you know, with the acid-fast stain, it's very specific, right? Very few organisms are actually going to come out positive. So for today, right, Same similar steps, right? It's a differential stain just like gram stain. So we're going to have a primary stain, which is going to stain all our cells, the carbofusion. We're going to decolorize, but in this case with acid alcohol. So make sure you grab the right one. Right? And it's going to remove the stain from the non-acid fast. So then we need to counter stain them and we use methylene blue for our procedure. Right? So notice even this picture I stole from somewhere, right? They had malachite green as the counter stain. As I said, it really doesn't matter what you use as a counter stain as long as it's not similar to carbofusion, right? So you're not going to get them confused. You wouldn't want to use a pink dye. So these are some examples of what they would look like. Like I said, a lot of times they clump together, right? So here's a big old clump of them. You want to scan around on your slide, hopefully, too, where you can see the individual cells. This is a different species of mycobacteria, right? Non-pathogenic, same like the one we're using today. We're obviously not using Mycobacteria leprae, are definitely not tuberculosis. Right? No pathogenic mycobacteria, but there are others that aren't. We are using Staphylococcus epididymis, so that's going to be your non-acid fast. Notice that they're mixed in together. They're in two separate circles for your guys' slides. So why do we do this? Remember, we want to know if they are within that genus, if they have those characteristics. We're going to perform our procedure correctly, hopefully. Right? We'll record our results. What do these results mean? Right? If they're acid fast, they have that mycolic acid in their cell wall. They're probably a microbacteria. If they're non-acid fast, right, they lack that mycolic acid. And therefore probably not a mycobacteria. Endospores, so remember we're using a particular method, the Schaefer-Fulton method. An endospore is a dormant form of the bacteria, right? Sounds like reproduction. They probably thought it was reproduction at one time. It's called endo because it actually forms within the cell. Right? And it does this under harsh conditions, and that could be any number of things, right? But definitely being without food, it would be a harsh condition, right? And that's the one we employ in this lab. But certain changes in oxygen or other gas requirements for organisms will stimulate them to form endospores. Depletion of water, just like for any organism you have to have it, are going to cause them to go into this state. And what they're doing is protecting their DNA. They're packaging it in such a way that it's protected so that when conditions become good again, they can germinate back into the original vegetative cell they were once before. A lot of the organisms that have this ability are saprophytes. There are decomposers. They eat stuff that was once living. And again, they're heterotrophic, meaning they're getting their carbon that builds their molecules from an organism that was once living, from organic molecules. You could put us as a saprophyte, right? So most of the bacillus that we are working with today, again, non-pathogenic, mostly ones that are in the soil or decomposers. Keratin is the main component of the spore coat that helps protect that protein we produce as well. It's in our skin that helps make it water repellent. The dead layer of cells that is on the outer layer of our skin. Our hair, our fingernails, right? That's why they have the hardness to them, is because of the keratin, that very strong. So there's a much stronger version in some animals that makes up their hooves, right? And their horns and shells of other animals, right? So that makes them really, really strong. Because of that really strong protein, we're going to heat them up, right? And I just thought of an analogy today as I was playing with my nails. Typically, some people like really hard nails, right? And so for me, I have really hard nails for some reason. So a lot of times, after I've taken a nice warm shower, is when I cut my nails, because they're softer because of the moist heat of taking a shower. So the same thing's going to happen to those proteins when we heat them today on our steaming pots. It's going to soften them and help drive that dye into the endospores so that we can visualize them under the microscope. So some pathogenic ones are Bacillus Anthracis, right? So the bad news, again, is these guys have the ability to form spores. And so that powder that went through the mail, that was millions upon billions of endospores. When a person inhales that, the endospore can recognize it's in a nutrient-rich environment, and it germinates. The bad news is this one is a very pathogenic bacteria, and it can kill you even with treatment. Okay. The good news is in the United States, we've done a really good job of cleaning this up from the environment. Animal carcasses or anything that's been contaminated with it have been properly disposed of. So it's not highly prevalent in the United States. We do not vaccinate against this infection in the United States. Other countries around the world, though, they do still have problems with this, right? So especially military that are deployed overseas get vaccinated against this. But that's also why it could potentially be used as a bioterrorist agent, right? Because we don't vaccinate the general populace because we shouldn't hopefully be exposed to it.
Is it difficult vaccination?
Not like smallpox is, you know. So another one that we do vaccinate against is Clostridium Tec9. And this one is, the major problem with this one is the toxin that it produces. So the toxin that it produces interferes with your motor neurons that tell your muscles to relax. It's your inhibitory motor neurons signaling your muscles that it interferes with. So your muscles stay contracted. And one of the common places where that would happen for individuals is in their jaw region. So this one is actually given the nickname lockjaw, right? Tetanus infection. You're actually vaccinated against the toxin this bacteria produces, not the bacteria itself. Again, because that's the major problem for this. So if you were to get an infection with it, you would still need to take antibiotics, right, to eliminate the infection, but you'd be protected by vaccination against the toxin, which is the really scary part. Every 10 years, right? The type of, and again, it's usually a wound type situation where you're going to get the infection, although sometimes you can get an infection of the gastrointestinal tract with this. These spores are highly prevalent in our environment. I don't think we'll ever be able to achieve to get rid of them. They're a soil contaminant. So a lot of times what people freak out is like, oh, I stepped on a rusty nail in my yard. The rust is not the problem. The rust is an indicator that it's probably been in your yard for a long time. Although in Louisiana, that probably isn't necessarily the case either. But a significant amount of time, meaning that it very most likely could be contaminated with these endospores. And they're microscopic. You're not going to see them. And then the type of wound a nail would create is a puncture wound, right? And so it's deeper than it is wide. And so the chances of an anaerobic environment from the damage happening in your tissues are pretty good. And this is an anaerobe. This organism is anaerobe. So under those wound type conditions, right, or any conditions where you're severe damage to the tissue, where you could have an anaerobic environment, is where these endospores would germinate, right, and then would produce the toxin that could potentially be extremely damaging.
Okay, so most people don't vaccinate every 10 years. They get it when they need it.
Right.
That's time for tour once you've already had it.
Yes and no, because most of the time it's one of those things like, I remember like when I was a kid, I got bit by a hamster when I was like 12, right? So chances are I probably needed my tetanus. And they gave me that shot before they even stopped my finger from bleeding. And meanwhile, like they're asking my dad my social security number, you know. He's like, I have insurance. Fix my daughter. You know, I was 12. I was like, I don't know my social security number. Who needs that information? But anyways, yeah. And again, you know, it's really going to depend on the situation. But yeah, a lot of times, you know, but it's very rare that people nowadays are going to get a serious, I would think that it would be rare, that people would get a serious wound and not get it properly taken care of. But again, people aren't very good at keeping up this vaccination. So when you have a wound, it's an opportunity to make sure you've been vaccinated. But if there's a chance, if they think that you may have a serious infection, like you've waited, you had a wound and you waited, they can give you antibodies from somebody else that will help you with the toxin. So there is a treatment that they can give you. So Clostridium Botulism is another one, right? And this one we worry about in our canned foods. And this is why in the canning industry, the way they do things, they do it a particular way. This is also why you never want to eat a swollen can, because chances are something grew in it. This is another anaerobe, so it grows under anaerobic conditions. But again, it's not the bacteria that's the problem, it's the toxin that it produces. And again, affects muscle contraction, right? Hence why people get derivatives of this toxin injected into them purposely, right? So that their muscles will not contract and they won't have wrinkles, right? I don't know why you would want to do that. And it does wear off over time, right? If it enters into the bloodstream, it can cause serious problems other places. It's not worth it. So notice this poor, unresponsive child because of ingesting the toxin. The last one, two more. Clostridium perfringens, again, endospore former, can cause problems. It'll eat away at your tissue. And again, this is wounds that have got dirt in them, not properly cared for, left unattended. You can get a particular type of gangrene. In this case, this is a gas producer, so you get these bubbly, frothy, oozing stuff coming out of these types of wounds. So other interesting fun facts for you guys to read about. Clostridium difficile is another one, spore former. Again, doesn't usually create problems unless individuals have a disruption of their normal flora of their intestines. And then this one can take over and, it can destroy someone's intestines. Also, nurses have told me, I've thankfully never experienced this, but apparently this one smells really bad, this particular pathogen. Like, before you even walk into the patient's room what infection they have. It's that distinct. So that's one you may run across. As I said, knowing the location of the spore would help in identification. Underneath the microscope, test-wise, right? I'm not obviously going to ask you. The spores themselves can be spherical, right? This one's probably the closest to a sphere, this one right here. The rest are pretty much elliptical. Good luck. I don't even know if it's elliptical or spherical. I think most of the ones we have are oval that you'll look at. Right smack in the middle, which picture?
Central.
Central, A. Terminal,
C.
C and
D.
D. See how both of those, right? This one, the spore is even bigger than the cell itself, so it creates it kind of swollen. And then subterminal is B, right? And our sphere, as I said, is D, and the rest are probably elliptical oval shape, clearly seen in this picture. So a vegetative cell is one that's actively metabolizing and reproducing cell dividing. A spore mother cell is when the spore is started to be formed within it. And then that spore can germinate and go back into the vegetative life cycle, right? Over time, the cell itself will deteriorate, and so you'll end up with just the spore by itself, right? What we call free spores. So again, it's the DNA, right? They make a copy of it, they package it up, they store it, because that tells the cell how to do things, when to do things. It's the command center, it's the information. So that's what needs to be protected, right, when they're under harsh conditions. The bad news for us is that you can't boil and destroy these, although the good news is when they tell you to boil your drinking water, it's usually not because it's endospore contaminated. It's contaminated with things that are destroyed by boiling, right? Most chemicals can't disrupt the spore coat and therefore deactivate these guys. Some forms of radiation can penetrate. UV light cannot. It can't penetrate past those layers. UV light, something as thin as sunscreen, helps protect you, right? It's not a very penetrating type of radiation. So for this, again, we're going to stain all the cells. You're going to make your bacterial smears. You're going to decolorize with just water. You're going to need to really rinse these slides good. Keep an eye on how much water you have in your trays and maybe dump it. Then you're going to counterstain with safranin so you can actually stain the cells. We might get lucky and actually see, the endospores inside the cells, but as you saw in mine, right, you probably see a lot of free spores too, but try and scan around and see if you can find any still in the cells.
In the video that we watched at the very end after she found her stain.
Yeah, she just blotted it. Yeah, that's fine. This one they're not going to wash off though, like with the capsule stain. So if you rinse it, it's fine. Either way is fine. But that was the inspiration for last time for not rinsing our capsule stains anymore. I was like, let's see what happens. It came out better. So why do we use old cultures?
Because they're more malnutrition.
Right, because we're depleting them of nutrition. We want them to form endospores so you guys can stain them. What does a positive result for the spore stain indicate about the organism?
That it lacks nutrients.
It's a spore former,
Spore form.
right? What happens if you don't see any spores? Does that mean that organism isn't a spore former?
No.
No, it means that you may not have employed the conditions in order for it to cause an endospore, right? To form that production. So you need to make sure, right? So a negative is not definitive when it comes to spore stain. Make sense? Right. It's a possibly negative. Possibly not a spore former. But definitely if it has spores, it is. So, why do we not need a negative control? What are we going to stain other than the spores?
The cell in the background.
We're going to stain the cells, right? So if you don't see any spores and you don't see any cells, then you probably did something wrong, right? So we don't need to do a definitely non-spore former, right? As a negative control. The cells themselves, the vegetative cells, will act as a negative control. Spores don't easily stain, right? That's why, because of that protein coat that's where we're going to employ the heat. We've already used one of these bacilli earlier, and we did a simple stain. And remember I said there were some things on your guys' slides that didn't stain real well, and I said those were endospores. And some of your inside the cell, it was clear, right? And I said that was an endospore because I knew the organism, right? If you don't know the organism, do you know it's a spore? No. You have to do the spore stain to confirm, because sometimes there are things that don't stain inside of cells. Right? So you'd have to do an endopore stain to confirm. Right? The only reason why you guys knew those were endospores is I knew the organism I knew was a spore former. So again, when you guys get your unknowns, if you see stuff not stained inside, it's a good indication you may have a spore former, right? But you're going to have to do your spore stain to confirm. Right? So what does it all mean? If it has a spore, it's definitely a spore producer, right? Has that keratin coating layer. If it doesn't, it just means it may not, right? Or has not yet. It's not a true negative. Unless you know for sure. So you guys, when you get your unknowns, we only come to lab once a week, right? So yours will definitely be starved. I will give you an old culture to start with, actually. So if it's a spore former, you'll see spores. At least you should. You guys ready to have some fun?